Good morning, dear students. In this session, in continuation of catalysis topic, let us discuss about kinetics of acid-base catalysis. In that, we are also going to study enzyme catalysis and Michaelis-Menton equation, bio-enzyme and industrial applications. The contents of this session includes this kinetics of acid-base catalysis, enzyme catalysis, bio-enzymes and industrial applications. Let us start with kinetics of acid-base catalysis. Acid-base catalyzed reactions takes place by transfer of proton from an acid to a substrate molecule, that is S, or from the substrate molecule to the base. Hence, the reaction between an acid, that is HA here, and the substrate S is as follows. Say for example, when substrate S is treated with an acid, that is HA, it gives rise to, in the forward reaction, that is SH plus A minus, that is a transfer of proton to the substrate takes place. It is a reversible reaction and the reversible constant is given by K minus 1, where this SH plus treated with a water, that is a hydrolysis, it gives rise to a product, that is P. As soon as the reaction is started, SH plus attain a steady state concentration. It attains a steady state concentration. Hence, the equation will becomes K1 concentration of S, concentration of HA is equal to K minus 1 concentration of SH plus into concentration of A minus plus K2 concentration of SH plus. Or this equation can also be defined like this, that is concentration of SH plus is equal to K1 concentration of S into concentration of HA over K minus 1 into concentration of A minus plus K2. This is equation number 1. The rate of the formation of a product is given by dP by dt is equal to K2 concentration of SH plus is equal to K1 K2 into concentration of S into concentration of HA over K minus 1 into concentration of A minus plus K2. This is equation number 2. So, here we have two cases. In the first case, if K minus 1 is very much greater than K minus 1 into concentration of A minus, then the above equation, that is equation number 2, gives rise to dP by dt is equal to K1 into concentration of S into concentration of HA. This is equation number 3. That is, the reaction rate is first order with respect to each of the substrate and acid molecule. The reaction for which the rate depends on the concentration of HA is to be subject to the catalysis. In the second case, if K minus 1 is very much less than K minus 1 concentration of A minus, then the equation 2 becomes dP by dt is equal to K2 into K1 over K minus 1 into concentration of S into concentration of HA over concentration of A minus. This is equation number 4. So, after invoking the equilibrium constant, the expression is like this. That is, Ka is equal to concentration of H plus into concentration of A minus over concentration of HA. Or this can be written like this, that is concentration of HA is equal to concentration of H plus into concentration of A minus over Ka. Then the equation 4 becomes dP by dt is equal to K2 into K1 over K minus 1 into concentration of S into concentration of H plus into concentration of A minus over Ka into concentration of A minus. dP by dt is equal to K2 K1 over K minus 1 into Ka into concentration of S into concentration of H plus. Thus, the reaction rate depends on the hydrogen ion concentration. Although the proton is initially transferred from Ha, that is an acid, in this case, the reaction is to be subjected to specific hydrogen ion catalysis. The base catalyzed reactions. What happened in the base catalyzed reactions? See, what happened in this reaction, it involves the transfer of proton from the substrate to molecule. Here it is from the substrate to molecule to the base. From the substrate molecule to the base takes place. If the rate of a reaction depends on the concentration of the base, here the reaction is to be subject to general base catalysis. For a reaction catalyzed by OH minus, the rate constant is K naught is given by K is equal to KOH minus concentration of OH minus. This is equation number 6. In general, if a reaction which follows the rate law, then what happened? D of concentration S by dt is equal to K into concentration of S. So it is equation number 7. And catalyzed by all the catalytic species, if it is catalyzed by all the catalytic species, say for example H plus, OH minus, HA and A minus, then the first order constant K in a buffer solution containing HA and A minus may be written as K is equal to K naught plus KH plus into concentration of H plus plus KOH minus of concentration OH minus plus KHA of HA plus Ka minus concentration of A minus, where K naught represents the rate constant of uncatalyzed reaction. Let us coming to the enzyme catalysis. What are enzyme catalysis? See, these enzymes are nothing but a complex organic molecules like protein molecules which are produced by living plants and animals. Say for example, dietase, gymase, etc. Here, the chemical reactions which are catalyzed by enzyme to form a product are called enzyme catalyzed product, and the process is called enzyme catalysis. Say for example, when the starch is treated with a water in the presence of a enzyme catalyst, that is a dietase, formation of a maltose takes place, that is C12H22O11. Another example, when maltose treated with water, again in the presence of dietase, the formation of glucose, 2 units of glucose takes place. Coming to the characteristics of enzyme catalysis. All enzymes are proteins and shown common properties here. Second one, enzymes are associated with chemical reaction occurs in the living system. Enzymes generally accelerate biochemical reactions by reducing the energy requirement. They reduce the energy required for the conversion of a reactant into product. Enzymes do not alter the amount or nature of the product. Here they won't alter the nature of the product or amount of the product. Enzymes do not affect the amount of energy released or absorbed during the reactions. Whatever the energy released or absorbed, they do not affect. Next point, enzymes retain identity at the end of the reactions. After the completion of the reactions, these enzymes retain the originality or identity. And the next point, enzymes extremely small amounts here and extremely small amounts of enzymes are able to bring about measurable changes. A little amount of enzymes are required in the reactions, and these enzymes are specific in their actions. Coming to the Michaelis-Menten equation. Here directly we are going to the equation, and it is given by R is equal to K2 into concentration of E naught into concentration of S over Km plus concentration of S, where Km is equal to K minus 1 plus K2 over K1. This is Michaelis constant, where K2 and Km are characteristic constants for a particular enzyme catalyzed reactions. And E naught represents the total enzyme concentration, and S here, concentration of S, represents the concentration of the substrate. Coming to the derivation of this Michaelis-Menten equation. The kinetics of enzyme catalysis consists of two steps here. First one is formation of enzyme substrate complex. When enzyme treated with a substrate, that is E plus S, in the forward reaction with rate constant K1, it gives rise to a complex that is ES, where K minus 1 represents the reversible rate constant here. And the second step is decomposition of the complex, which is formed in the first step, that is ES. It is decomposed, gives rise to P plus E, that is product plus enzyme. So the rate of appearance of the product is given by D into concentration of P by T dt is equal to K2 into concentration of ES. This is equation number 1. Applying the steady state approximation to ES, it is given by D into concentration of ES by dt is equal to 0 is equal to K1 into concentration of E into concentration of S minus K minus 1 concentration of ES minus K2 concentration of ES. So ES is given by concentration of ES is equal to K1 concentration of E into concentration of S over K minus 1 plus K2. So ES is equal to concentration of E into concentration of S over Km, where Km represents K minus 1 plus K2. Observe here, Km represents K minus 1 plus K2 over Km. You can observe here. In the previous equation, we have K1 by K minus 1 plus K2. This is what represented by Km, that is K minus 1 plus K2 by K1. This is Michaelis constant. So, if E naught is the initial concentration of the enzyme, then at any time during the course of a reaction, it is concentration of E naught is equal to concentration of E plus concentration of ES. So ES is given by concentration of ES is given by E naught minus E. So this is equation number 3rd. So from the equation 2 and now the equation 2 becomes E naught minus E. Observe the equation number 2. E naught minus E is equal to 1 over Km into E minus concentration of S. So E naught is equal to concentration of E into 1 plus concentration of S over Km. Or this can also be written like this, that is concentration of E is equal to concentration of E naught over 1 plus concentration of S over Km. This gives rise to Km into concentration of E naught over Km plus concentration of S. This is equation number 4. So the overall rate expression given by like this, that is R is equal to D into concentration of Es over dt is equal to K2 into concentration of Es. This is equation number 5. So from equation 2 and 5 we can write like this, that is R is equal to K2 into concentration of E into concentration of S over Km. From equation number 2 and 5 you can observe those two equations, that is 2 and 5 before writing this equation. So but from 4, equation number 4, we have R is equal to K2 into concentration of S over Km into Km concentration of E naught by Km plus concentration of S. So R is given by, that is K2 into concentration of E naught into concentration of S over Km plus concentration of S. This is what is called as Michaelis-Menten equation. So now let us come to the bio-enzymes. What are bio-enzymes? The enzymes catalyzing biological reactions. Okay, enzymes catalyzed biological reactions are called bio-enzymes. Examples: invertase and zymase are used in the production of alcohols. See, in the production of alcohols, okay, we are using zymase as well as invertase as an enzymes for the catalytic purpose. So this is one important example, and another one is lactic and butyric bacilli are used in the production of butyric acid. The formation of a butyric acid, these are acts as a enzymes for the catalytic purpose. Coming to the application for part of this bioenzymes, that is industrial applications of bioenzymes. Invertase and zymase enzymes are used in the manufacture of ethyl alcohol from molasses. This is what a familiar one, that is a fermentation process. Here we are using invertase and zymase. Second one, diastase and maltase are used as a catalyst in the manufacture of ethyl alcohol by fermentation of starch. By the fermentation of starch, we are using diastase and maltase. And next point, Mycoderma aceti acts as catalyst in conversion of alcohol into acetic acid. In the conversion of alcohol into acetic acid, we are using Mycoderma aceti. The next one, lactic and butyric bacilli are used in the manufacture of butyric acid, as we have discussed earlier. That is, it's also a fermentation process. Lipase convert cane sugar into glycerol. The enzyme lipase convert cane sugar into glycerol. And another one, lactic bacilli converts milk to curd. We know this conversion of milk into curd. The lactic bacilli acts as an enzyme here, lactic bacilli. And the next one, urease converts urea into ammonia and CO, that's carbon monoxide. Urease. These are the important industrial applications of bioenzymes. So coming to the revision of these sessions, we have discussed kinetics of acid-base catalysis in detail. Later, we have also discussed enzyme catalysis. In that characteristic of enzyme catalysis, Michaelis-Menten equations, and we have also discussed about bioenzymes and industrial applications. These are some of the references for this session. Thank you.